So I'm really happy to introduce today's distinguished speaker. Today we have Pei-Yong Shi, who is John Seeley Distinguished Chair in Innovations in Molecular Biology at the University of Texas Medical Branch in Galveston. He is an elected fellow of the American Academy of Microbiology, adjunct professor of emerging infectious diseases at the Duke NUS Graduate Medical School in Singapore, and honorary professor at the Wuhan Institute of Virology at the Chinese Academy of Sciences. He works on RNA virus, drug discovery, and vaccine research. His lab's research focuses on viruses that cause significant human diseases such as dengue, Zika, West Nile, and SARS-CoV-2. His group developed the first reverse genetic systems for the epidemic West Nile virus and Zika virus, and they discovered flavivirus N7 and 2'O methyltransferase activities. And today he's going to be presenting, I believe, some recently published work on neutralization of SARS-2 variants by the Pfizer vaccine and engineering an IgM neutralizing antibody. So Pei-Yong, please take it away. Sorry, Pei-Yong, you're on mute.
Thank you, Spencer. It's really a great pleasure to talk about our work. As Spencer introduced, I'm not the coronavirologist by training. As all of us here, we just feel obligated in response to the pandemic. So that's we pivoted towards the COVID research. What I'm going to do today is just quickly to introduce what we have been doing. And a lot of them is through collaborations with groups, not only on the UTMB campus, but also with external partners, including major pharmaceutical companies. I will first start to talk about our genetic system we developed for the SARS-CoV-2. And then we will start to talk about those tools, how we use these tools to screen for antiviral inhibitors, and then to help with the vaccine development, particularly with Pfizer's vaccine. And then lastly, I will talk about the new variants and their potential on the neutralizing antibody activities of the vaccine. It is not advancing. Okay, so good. Can you see my slides? Just want to make sure. Okay, good. So when we initially entered the SARS-CoV-2 study, we thought based on our strength on molecular virology, we started to develop the reverse genetic system for the virus. As you know, the virus is a single-stranded RNA of +sense polarity. And because it's 30 kilobase long, what we did is we performed RT-PCR using 7 fragments to cover the whole genome. And then we, in the test tube, to in vitro ligate those 7 fragments together. And then that represents the complete cDNA copy of the genome. And then on the 5' end, we engineer T7 promoter so that we can do in vitro transcription to in vitro synthesize the RNA. And then when we transfect these RNA into cells, and then we're able to recover virus. As a virologist, this is the most important tool that you would like to study the virus, because that allows you to make any changes on the genetic footprint, any amino acid, and you will see the power of this platform. And we published the first peer-reviewed reverse genetic system for the SARS-CoV-2. And then using this system, we very rapidly to engineer so-called reporter virus. So what we did is we engineer reporter genes, in this case indicated here, luciferase, into the viral genome, specifically into the open reading frame 7 of the viral genome, and we can recover the virus as it's shown here very robustly. And then if you infect the cells with this virus, it will create luciferase. It's just like identical the wild type virus, except when it infects, it expresses this extra gene, and that allows you, as indicated here, to look at over the time of infection. You can see the luciferase signal on the y-axis, and that is very robust. And using this, that allows you to do the high throughput drug screening, so that you don't have to use the conventional way of plaque acid, which is very tedious, very low throughput. So using these platforms, we've collaborated with multiple companies. Here, I'm just showing you the collaboration with Gilead Sciences, and using the reported luciferase, we're able to very rapidly to pour out remdesivir from their clinically approved nucleotide drugs, which have been used in human or in clinical trials. And in the interest of the time, I'm not going to go into the other class of compounds, and that this, just to illustrate, this is very high throughput that can allow us to do antiviral drug screening. And then this is our recent work, I think published 2 weeks ago in Nature. And this is a collaboration with the UT Health Science in Houston. And we are developing a nasal deliverable, the IgM format for potential COVID therapy. So the whole story started with, we have identified some monoclonal antibodies using the phage display. And then one of the antibody is most potent. It's called COVID-2, this 14 antibody. And then this is just shows you the receptor binding domain, and the red string indicates the receptor binding motif. The green dots indicates the amino acid involved in this epitope of this antibody 14. And the idea is we want to convert this into the IgM format so that you will have more arms to interact with, to bind to its target spike. And then we think that might also overcome the resistant profile, as well as to increase its spectrum to inhibit the ongoing variants, the emerging variants. So as in panel C, you can see the EC50 of the original IgG format versus the IgM format. It increased the EC50 from 1.6 nanomolar to 0.8 to 2.8 picomolar. So you can see a significant potency increase. And then also we show this IgM converted form of the antibody overcomes the resistance, as well as it's has a broad spectrum against the different newly emerged variants. And in the interest of the time, and I'm not going to go into the details, you can see cross in the IgM, the neutralizing activity, the EC50, PINT50 is very similar against the different variants, P117, P1351, as indicated here. In contrast, the IgG, the same antibody, it loses its activity against the new variants. And then we also looked at the pharmacokinetics of nasal delivery of these IgM antibodies in the mouse model. As indicated here, you can see the antibody after the single dose nasal delivery, it's quite sustained to a high level all the way to just a single dose to the 96 hours post dosing. We can still see a significant level of the IgM antibodies in the nasal cavity. And then more importantly, in the efficacy mouse model, we can see the IgM, just a single dose, either prophylactically treated the mice before the infection, or after the infection, treat with a single dose of this IgM antibody, single dose, it significantly reduces the viral load in the lung respiratory tract, lung loading of the viral infectious viral RNA. So if you're interested in this, just go refer to the original paper. And we're very excited that this it has been licensed to the IgM Sciences, and we're going to deliver that and trying to enter into the clinical trial. So then besides that luciferase assay, we also engineered so-called the fluorescent labeled viruses indicated here using the M-neon green, which is the green fluorescent reporter we engineered at the same location, ORF open reading frame 7, and we can recover the green virus. So this is very handy for neutralizing assay, which is highly sought when all the vaccine companies were developing their vaccine, because as we know, one of the key criteria for immune protection is the level of the neutralizing antibodies after vaccination. So the assay is very simple. It can be done at the 384 format or 96 well indicated here. Day one, you see the uninfected cells. And then the next day, you mix your tested serum you're interested to mix with the virus for 1 hour. And then you lay the mixture onto the preceded cells. 16 hours later, you just use the high content imaging to measure the green cells, the inhibition of the green cells. And that will allows you very rapidly to calculate the neutralizing antibody levels. And then using these assays, we partnered with Pfizer. So Pfizer's preclinical and phase 1, phase 2 neutralizing activities were all done using our assay, and it's done at the UTMB. So we're very happy to be able to contribute to that. And then the same assay as indicated here has been used by Moderna to look at characterize the vaccinated individual. Indicated here is a 6-month durability of the neutralizing activities published in the New England. You can see 1 dose, 2 dose at the 3 different groups, age groups. You can see over this time of 6 months, there is a drop of the neutralizing activity. But even that after 6 months, the neutralizing activity is still above 100. So it still has a high level of the neutralizing activity. So all these.
Pei, do you mind if I ask a quick question?
Sure.
Just on your previous slide. Sorry, I'm not an expert or anything in this, but could you explain the y-axis here and what unit that is and what exactly it's measuring?
Okay, so this is the neutralizing antibody titer. So basically a specimen you dilute it to what fold until you will lose 50% of its inhibition. That is the number as what we are plotted here.
I see. Thank you.
Sure. And then all these are very good, high throughput, but there is one limitation of these assays I just presented, which it requires by safety level 3. So a lot of labs, particularly industry, they don't have access to the BL3. So the question is, can we develop an assay that can recapitulate the authentic viral infection, but also can be done at the BL2? So we recently published this system in Cell, and the system conceptually allows us to do that. And very happily, I would say that 2 days ago, NIH also approved that we are able to use this system at the BL2. The UTMB IBC, Institutional Biosafety Committee, already approved. So we're now be able to move this system into the BL2. So the system works in this way. What we did is we delete 2 genes using engineering of the reverse genetic system, delete 2 genes, in this case, of 3 and the envelope. And then we created a transcomplementing cell line, which inducibly express those two missing genes inside the cell. So when you electroporate or transfect these RNA without the two genes into these transcomplementing cells, and then you will be able to recover particles, right? Viruses. And this viruses is identical to the wild type virus, except when it infects normal cells, it will start to infect and then translate viral proteins and replicate its RNA. But because in these normal cells, it misses those two genes, and then it's stuck. It cannot produce the progeny viruses. And that's the concept. You can do it at the BSL-2. So using this system, we demonstrate this system because we also engineer reporters into the system. That allows you to do high throughput antivirus screening, neutralizing activity for vaccine development. And now we are collaborating with our hospital team, trying to adapt these for the patient service to look at the neutralizing antibody levels. So now I'm going to talk about the variants. I'm going to talk about two topics of the variants. The first to talk about the biology of the variants. And then I will talk about the neutralizing activities with the new variants, also including the Delta variant we discussed earlier. So for the new variants, as you know, that initially the first prominent mutation after the outbreak or after the pandemic, it is this spike mutation D614G, which was early detected in the February of last year. So it is we're very interested in analyzing what does this mean? Does this mutation increases the fitnesses or transmissions, and how does that affect the vaccine efficacy? So what we did is we used the reverse genetic system to make a pair of viruses we call isogenic viruses, everything identical, using the backbone of the Washington isolate, the index case isolated in the US. We changed the original D614 to the G614, only one amino acid differences. And then once we recover the viruses, as indicated here, the plaques are very similar between the two viruses. And when we grow them on the Collo3, which is the human lung epithelial cells, you can see the mutant virus indicated in red has a slight advantage. And then using the hamster model, we compare the viral replication in the respiratory tract between the two viruses. We intranasally infect the hamsters with either the original virus or the mutant virus, and we monitor the weight loss as indicated in panel B. You can see both viruses caused an equivalent level of weight loss, indicating that the mutation doesn't seem to change the pathogenesis, at least in this model. But when we measure the viral loads in the respiratory tract, as indicated here in day 4 post-infection, those animals, you can see the nasal wash, the wild type in black, the mutant in red. There is over 10-fold differences. The mutant has 10-fold more of the infectious viruses in the nasal wash, as well as in trachea, the upper respiratory tract. In contrast, the lower respiratory tract, the different lobes of lungs of the infected animals, the mutation does not seem to affect the viral loading over there. So these data clearly demonstrate that the single mutation that enhances its replication in the upper respiratory tract that might potentially increase the transmission. So to further validate these hamster model, we use the human primary airway culture. So what we did is we infect those cultures and look at the viral replication in these cultures. So you can see in panel B, the mutant virus grows significantly higher compared with the wild type. And we also did the competition experiment So what we did here is you mix 1-to-1 ratio, the mutant and the wild type as a mixture to infect the human, primary human airway culture. And over the time, you measure the ratio between the mutant versus the wild type. So if a ratio is above 1, that means the mutant virus wins the competition. As indicated here, over the 5 days, the mutant virus wins the competition compared with the wild type. And we also, you can do 9-to-1 inoculum. That means 9 portions of the wild type virus versus 1 portion as a starting inoculum of the infection. And then over the time, you can still see the mutant virus wins the competition. So these data demonstrate that in human primary airway culture, that the mutation increases the replication fitnesses of the SARS-2. And we also look at the stability of the viruses, the infectivity over the time when you incubate the virus at 42 degrees, 37 degrees, and 33 degrees, which mimics the upper airway temperature. So you can see over the time, the red virus, which is the mutant virus, retains a higher infectivity compared with the original wild type virus. So these data indicates that this mutation also improves the thermal stability of the viruses that might contribute to the enhanced viral infectious particles in the upper airway. And then after we published our results, 2 other groups, Shi, Colloquium, and Ralph Berry, as well as Dave Wentworth, Volker Thiel, and Martin Beer's group, they used the real animal transmission experiment to demonstrate that this D614 clearly increases the transmission in the animal model. So that in terms of mechanism, structural biologists show that because of this mutation, and it promotes the receptor binding domain indicated here to an up conformation that is required to bind to the human S2 receptor, and that promotes the infection. That might be the underneath the mechanism of this mutation. And now the second study I want to mention is the B117. The reason we were interested in that is because it is predominant even now in the United States. So we are interested in whether this the B117 has more transmissibility using animal model. And then if so, we would like to identify which mutations in the B117 that is responsible or plays a major role to increase its transmission. So what we did is, again, using the genetic system, we have a Y type, which is using the Washington isolates, and then compare in the spike protein gene, the B117 has 8 mutations extra, in addition to this D614G, which I just described. And there are 8 extra mutations. So what we did is we made 8 mutant viruses individually containing single mutations in the background of D614G. And then in addition, we made another mutant called 8X, which contains all the 8 spike mutations into the virus. So we can recover virus indicated here. In the interest of the time, I'm not going to go into the details. So what we did is we use the hamster transmission model. So what we do is we infect the hamsters intranasally, and then on day 1 post-infection, we co-host a clean hamster in the same cage for 6 to 8 hours. And then we separate these 2 animals. And then we measure the transmission of the viruses in the respiratory tract of the virus, or in the 2 animals, the donor animals and the recipient animals. In the interest of the time, I'm not going to go into the details. So what we found is this mutation 501 in the receptor binding domain is the major determinant to enhance the transmission of the B117 in the hamster model. And then also in the same experiment, we tested in the human airway culture. Again, using the competition experiment I introduced before, you can see the 501, the single amino acid chain, significantly allows the virus to overcompete the original virus. In terms of mechanism, we showed that once you have the 501 mutation, that the receptor binding domain, the affinity to its binding receptor, ACE2, increases by more than 100-fold, as indicated here by the biophysics KD, K on, K off. So then for the last two slides, I'm going to talk about the neutralizing activities on the different variants. We took a very different approach compared with the other groups to look at the neutralizing activities among different variants. So what we do is we make so-called chimeric SARS-2 variants. We use the original Washington isolates. This is the original Y type we call. And then what we do is we swap the whole complete spike gene with the different variant. So we systematically follow the different variants when it comes. Once we get the sequence, we synthesize that, and then we drop into the original virus background. So now we can recover viruses. They are all SARS-CoV-2 virus. They are identical, except the spike genes has been completely swapped to the new variants. So that's how we prepare our viruses. And then in terms of the serum specimens, we collaborate with Pfizer and the BioNTech. We use the same 20 serum from the human who have been vaccinated with the Pfizer-BioNTech vaccine in the phase 1 and the 2 clinical trial. So we use the identical 20 set of patient serum so that historically we can compare the data among different variants. So that's the experiment we set. So this summarizes all the data we have over the past 6 months. So let's look at the bottom panel first. So for those 20 serum, each individual serum, we test them for their neutralizing antibody levels. So this is called a PINT, neutralizing 50% neutralizing antibody levels. The first bar is the Y type, the Washington isolate, and on the top is the geometric mean of the neutralizing antibody levels. The higher, the better, right? So this is the next one, is B117 alpha. You can see that it's similar, not much drop in our system. And the P1, and then you can see the B1351 has the biggest drop, which is about, threefold drop in terms of neutralizing activity. And then on the right, bottom right, you can see we have the B1 427, 429, which is the California, and B1 526, originally identified in New York, and then also a subtype, the B117, plus the E41484K mutation, which is important. This mutation has been identified to confer resistance to many monoclonal antibodies, including those have been approved for the clinical use right now. So you can see those variants didn't seem to drop the neutralizing level to a biologically relevant degree. And then this on the top is the more recent data, which is the Indian variants. And we also included the Nigeria variant, B1525. Among the different Indian variants, the B1617.1 is the most concerning one in terms of dropping the neutralizing activity. It's about threefold reduction. And then the others, the more prevalent one is the B1.617 version 2.2. And then also, we also did the B1.618. And the other three Delta variant didn't seem to drop the neutralizing titer too much. So I think among the Indian variants, the B1.617.1 in terms of neutralizing activity is of most concern. So I think I'm going to stop here to acknowledge the team and the great colleagues at UTMB to work together. And I mentioned the collaboration with Pfizer, BioNTech, the IgM antibody has been licensed to the IgM Bioscience, and then the Gilead collaboration for the antiviral, and then collaboration with Dr. Zhichang An at the UT Health at Houston, and Mike Diamond at WashU. We collaborate on the animal models, and then the funding agency. And I will stop here to answer any questions. Thank you.